Hola, bienvenidos todos. Es realmente un gusto para mí inaugurar los seminarios presenciales del Departamento de Fisiología después de casi 2 años. Y estoy feliz de verlos reunidos aquí de nuevo y espero que sigamos en este camino. Es además mucho más motivo de orgullo para mí inaugurar este seminario, considerando que nuestro primer expositor es un científico importantísimo y sobresaliente, el doctor Lars Chttka. La doctora Quintanar nos va a hablar un poquito más de él. Yo solo quiero comentar 2 cosas. Una, por favor compren su libro. Yo traté de comprarlo ayer, pero apenas va a salir publicado. Y les recomiendo que lo compren porque una cosa que sí hice ayer fue leer 3 de sus trabajos. Y yo he estado trabajando aquí desde hace 33 años, he leído muchos trabajos y realmente leer trabajos tan bien escritos como los del doctor Lars Chttka son muy pocos. Es de los mejores escritores de artículos que yo he leído en mi vida. Y se los digo sobre todo a los estudiantes de los últimos semestres, porque sé que están en problemas de escribir su tesis y sus trabajos de publicación. Entonces, si quieren un buen trabajo de publicación como ejemplo, lean cualquiera de los trabajos del doctor Chttka. So, it's a pleasure and a honor to have you here. You are going to inaugurate the presential seminars of the Department of Physiology. And Dr. Quintanar is going to tell us something about you for all the people here present and all the people that is listening to us through the official channel of YouTube of the Simvestar. Please, Doctora, come here.
Muy buenas tardes a todos. Yo sé que el profesor Lars entiende un poquito de español, así que voy a platicarles un poquito de él en español y no es nada que él no sepa. Su formación es en biología. Él estudió en la Universidad Libre de Berlín, hizo ahí también su doctorado en biología y después hizo varias estancias postdoctorales, una de ellas también en la Universidad Libre de Berlín y en la Universidad Estatal de Nueva York, Stony Brook. Después, por varios años, fue profesor docente en la Universidad de Würzburg y desde 2002 está en la Universidad Queen Mary de Londres, donde, pues además de ser profesor, desde el 2008 es director científico del Centro de Investigación de Psicología. Y de hecho, él es uno de los fundadores de este centro de investigación. Y bueno, pues su CV es larguísimo, no pretendo aquí leerlo todo, solo compartir con ustedes que ha sido editor de la sección de biología de la revista PLOS Biology desde 2004. También está en el editorial board de, lo voy a leer en inglés, del Proceedings of the Royal Society of London y en el Quarterly Review de Biología. Es también miembro de los miembros de Faculty of 1000, o sea, uno de los profesores de 1000, y también es chair del ERC panel. Ha recibido diversos reconocimientos por su trabajo, entre ellos solamente voy a mencionar algunos. Ha recibido el Royal Society Wolfson Research Merit Award, y también ha sido electo miembro de la Linnean Society of the Royal Entomological Society y también de la Royal Society of Biology. Ha recibido también el premio Leslie Goodman Award que otorga la Royal Entomological Society, eso lo ganó en 2006. Y bueno, pues es un autor muy prolífico, tiene un índice H de 53, para quienes importen de los números, pero más importantemente, el profesor Lars, su investigación está en la interfase de la fisiología sensorial, la psicología de aprendizaje y la ecología evolutiva. Principalmente se pregunta por qué los animales tienen los sistemas sensoriales que tienen y cómo los utilizan en el ambiente natural en el que viven. Y pues con esto los dejo con esta interesante plática de la mente de una abeja. And thank you very much, Lars, for joining us and starting these hybrid seminars at Simbestav. It's probably the first one on our campus. We're very happy to have you here.
Muchísimas gracias. Pero voy a hablar en inglés, porque mi español es muy mal. Lo siento.
Puedes levantar el micrófono un poco.
¿Sí? ¿Así está bien? Okay. All right. So it's a great pleasure to be here and talk to you guys in person. It's not the first talk for me after the lockdowns, but one of the first. It's only the first one was a month ago, and this is basically the second. Both of them were in Mexico, by the way. The last one was at UNAM. So it's wonderful to be here and wonderful to talk to you guys in person. It's a pity that Fanis Miselis, your colleague, couldn't be here. He initially invited me, and he's a former colleague in London. And I very much regret not meeting him here today, but we'll catch up soon. All right, so thanks for being here. It's really a great pleasure and privilege. And because of your kind introductions, I turned as red as a jitomate, I'm sure. But thank you very much. So my story with Mexico City is a funny one. This is me just 3.5 years ago. I looked much more well-groomed, I guess. And then I met a very nice lady in Coyoacán, and I stayed here most of the time since. Given how much time I've spent here, it's actually embarrassing how bad my Spanish is. But yeah, this was me giving a talk at UNAM just 3.5 years ago. So what I want to talk about today is the question of whether bees could be said to have a mind, and that might seem counterintuitive to most of you, that we would credit as little a thing as an insect with having a mind or consciousness. So I'll talk you through the ideas that I have in this context, and I'll focus specifically on spatial representations in the mind, on how bees represent space in their brains, how they think of things in space in the context of space, whether they can foresee the outcomes of their own actions, and so in a sense look into at least the immediate future. And I want to explore whether they attach emotional states to spatial settings. By the way, if I ever speak too fast or you have any questions, just raise a hand and interrupt me at any point. So I think many of us are aware that insects can in principle do very impressive things. So we are aware of their rather remarkable architectures that bees and ants, wasps and also termites can construct. And here's an example of two structures that look perhaps superficially similar, at least if you subtract away the size. Then these termite hills that are actually called Cathedral termite hills look superficially similar to the Sagrada Familia in Barcelona. But the general thinking is that these structures in humans and insects come about by entirely different processes. That in humans there is a top-down organization where there's an architect who plans the whole thing and then gives instructions to all the workers, whereas conversely people tend to think that in insects there is no guiding mind, there is no planning, and instead everything, all these impressively complex structures, arise by self-organization, by simple rules by which individuals interact with using innate behaviors, and the whole structure emerges just from these simple local interactions. And I want to challenge this point of view a little bit and to ask whether insects really have no form of spatial planning, both in their constructions as well as other activities. Now, the question of whether in bees and other insects everything arises by self-organization is actually older than many people think. So here is a text from Charles Bonnet, a Swiss naturalist who was impressed, like many scholars, with the architecture of honeybee wax comb. So you can see this magnificently regular and symmetrical structure of a honeycomb on the bottom left. It turns out it's actually a mathematically optimal structure in terms of material used for storage space generated. It's also double-sided, so the efficiency in generating storage space for honey as well as bee larvae is certainly very impressive, and so is the high regularity of the structure. And Charles Bonnet said this about self-organization and actually consciousness. He said, place together in the same room 10,000 automatons, animated with a living force and all induced through the perfect resemblance of their outer and inner being. If we admit the least degree of feeling in these automatons, even only such as is necessary for them to be conscious of their own existence, seek their own conservation, avoid noxious things, prepare useful things, etc., their work will not only be regular, well-proportioned, similar, equal, but it will also have symmetry, strength, convenience to the highest point of perfection. So interestingly, Charles Bonnet thought that emotions and consciousness were almost at the basis of the process, and that just from placing all of these individuals that are equipped with feelings and emotions and so on, by self-organization, something as magnificent as the hexagonal honeycomb structure will arise. Now, we now know that it's clearly not that simple. Humans have had all kinds of cruel experiments in battery farming and so on, with putting tens of thousands of individuals into tightly confined spaces. And clearly, any sort of regular structure does not just arise naturally out of putting lots of individuals into the same room. So a little bit more than that is certainly required. So the question of whether comb constructions, these you can see the formation of natural honeycomb here in an observation. Beehive have been replaced with glass screens, and you can see that the process typically starts at the top of the hive, and then bees gradually work down along the direction of gravity to expand their construction, and they often form these strange hanging chains in the process of building combs. And François Huber, another Swiss naturalist, was the first to explore this process both in terms of observation as well as experimentally. And so he experimented just because he wanted to see the inner workings of a beehive by first of all replacing walls of the hive with glass screens so that he could look inside. And what he did first was to put a glass ceiling into the hive and then to observe what the bees did. And one of the many things that they did was they stopped attaching their growing honeycomb construction to the ceiling. It turns out that they did not like glass very much. And what they did instead in that case is they built the entire comb construction as a tower from the bottom up rather than from the top to the bottom. Okay? So he was already very surprised 200 years ago by this flexibility. And it's not trivial if you think about it, because if you were to build a robot that simply mimics how bees do this construction naturally, that is using gravity as a reference, starting at the top and building their way down, that robot would already fall flat on its face if it could not attach its construction to the ceiling. So bees just inverted the whole procedure and built a bottom-up construction. So Huber next thought, all right, so now what happens? if I replace both the ceiling as well as the floor with glass? And in that case, what the bees did is they started the construction attached to one of the side walls and built the comb through the cavity early. So again, there was another degree of freedom by which they could adjust this construction. But the really surprising thing, and again, you have to think these are 200-year-old experiments, so it's quite remarkable what the researchers did there. So after the bees had already begun a construction through the cavity of the hive, a lateral construction, Hubert and his team then put a glass screen into the path of the ongoing construction. So that's shown here on the bottom in panel B. So the target wall against which the growing comb was going to be attached was now rendered suboptimal by putting a glass screen there, to which the bees did not want to attach their comb. And what happened in that case was that before reaching the suboptimal outcome of attaching the comb to a slippery surface, is that the bees turned the entire construction by 90 degrees, building a curve into the comb and attaching it to the nearest wood wall. Okay, so it seemed to Hubert, and these experiments remain to be replicated, the bees took corrective action way before they had reached a suboptimal outcome of attaching the comb to a slippery wall, before actually realizing, oh, this does not work out well, and instead avoided this suboptimal future outcome to turn the comb and attach it to the nearest wood wall. And he said at the time, in 1814, I acknowledge that I could not suppress a sentiment of admiration for an action in which the brightest foresight was displayed. Now, these experiments remain to be replicated with large sample sizes, as we would expect them in the 21st century, and one of my PhD students is currently looking into that. But it's a very impressive early exploration of whether bees simply followed hardwired rules that allowed them to build their constructions as they will under natural conditions, where they always start at the top and always build bottom down, top to bottom, and are faced with challenges that until then in the entire evolutionary history of bees, a novel challenge for bees to deal with, and they dealt with it in remarkable ways. Now, most of the work that we do nowadays on the intelligence of bees is not inside the hive, but outside. And we capitalize on bees' natural lifestyles, which involve quite a bit of learning. So bees, and that is not just honeybees, but also the many thousand species of solitary bees and stingless bees, of which you have quite a diversity in Mexico as well. All these bees have a home. They have a place where they build their nest, where they rear and provision their young, and which they must therefore remember. Okay, so they have to remember the place where their nest is, because if they don't, then all their efforts towards collecting nectar and pollen are lost for the colony. And if they're solitary bees, which are all single mothers basically, if they forget where their home is, well, they lose all their babies. Okay, so their selection pressure to have a highly precise spatial memory is very important. Moreover, as flower foragers, they need to be essentially careful shoppers in the flower supermarket. They need to evaluate often dozens of different flower species for their nectar and pollen offerings, remember them, and remember to associate with the rewards they've found the signals that make up the flowers. So they have to remember perhaps that the blue flowers are more rewarding than the yellow ones, or that the yellow ones have more pollen and the blue ones more nectar, and so on, out of the dozens of different offerings that are out there in any bee's flight range. So they have to make these comparisons and learn about them. And we exploit this readiness of bees to learn about visual signals, for example, in the laboratory, because one remarkable thing is that bees, unlike other laboratory animals, never satiate. Once their tummy is full, They go home, regurgitate the food they've collected, and come right back to the flowers, or in our case, to our setup. And on the left there, you can see one of our perhaps more unusual experiments where we trained bees to associate images of human faces with rewards. And you can see the honeybee is about to land on a little platform that has a droplet of nectar. So bees' flexibility in associating visual signals with rewards is quite great. There's no evidence that bees recognize what we're giving them there as a face. They just recognize it as a visual pattern that is associated with reward. So in a bee's view, this is probably just a very strange flower. Now, navigation in human-built environments is made relatively easy by the fact that humans build, at least in cities, landmarks that are designed to be unique and memorable. So even if you have to find your way around a city, this is Shanghai, I think, where you've never been, you don't understand the language, and even if your mobile phone is out of charge, you can use these landmarks because they're built to be memorable and navigate by them. Now, conversely, a bee's natural environment is something like this, where the hive might be, or the nest under this tree there on the right, or in the tree, and there is, let's say, a pollen patch that's over there on the left, where the bees can find a useful protein source, and another flower patch that is valuable for nectar might be a kilometer behind the hill in the back there. Now, I don't know if any of you hike. I like walking quite a bit in the cerros around Tepoztlán, and people get lost there all the time because it's actually very difficult. It's very difficult to navigate through a pathless environment with no visible landmarks. And if you have no mobile phone reception and perhaps no physical compass, it's a very tough challenge. And you have to imagine that bees, honeybees, for example, fly over 10 kilometers away from their native hive and reliably find their way back to the point of origin. So the way we study this navigation nowadays is by a technology called Harmonic Radar, where, so here you can see our equipment rotating with two dishes, and the bottom one basically emits a signal that is picked up by a transponder attached to a bee's back. This is a bumblebee, an abejorro, with a transponder on the back, and this little antenna-like device bounces back a different frequency, that's why it's called a transponder, that is subsequently picked up by the top radar dish. And using the delay of the signal and its directional signature, we can determine the precise position of the bee as it navigates, and we can do that over their entire lifetimes. And the system that we've recently developed to some extent allows us to track not just coordinates, but also the height at which the bees fly. And using such technology, we can not just reconstruct where the bee is, but we can also take the view from the bee's cockpit, if you wish, and reconstruct the bee's view as she navigates around the landscape. This is a track of a male honeybee, by the way, and you can see these sort of boomerang-shaped things are their eyes, which are curved, and they can look pretty much all around. Their spatial resolution is relatively poor compared to our own. You can see it's very pixelated. They compensate for that to some extent by being able to process more information per unit time than we can. But yeah, so this technology allows us to reconstruct what the bee sees while she's flying, and therefore to explore how they're strategically using landmarks to facilitate their navigation. And among the many challenges that a bee has to face is not just that she has to remember where the hive is and where one food patch is, but also often to link multiple destinations in a way that minimizes travel distance and energy expenditure. And that is what's known in the human world as a traveling salesman problem, where you have to link a variety of different destinations. Let's say you're a sales representative for a publishing company and you need to think about ways in which you can minimize travel distance by, if you're having to link multiple Mexican cities, then of course if you don't carefully calculate the sequence in which you visit the cities, then there's a risk of you spending a lot on gasoline or airplane tickets depending on how you travel. So here are 5 destinations, each marked in blue on the left, and the hive is at the bottom. And you can see a radar track of an individual that in this case is not yet very good because the bee has not yet had much experience. But if you do this multiple times and allow the bee to explore where these locations are and to find a better route, then she gets better and better. So here's such a track of an individual bee. The bout number is shown at the top, and you can see now she's found the first few, now the second 2 destinations, now the 5th one. We're in about 9 at the time now. And as the bee progresses, she's still doing quite a bit of exploration, but the trajectory gets more and more streamlined until in the end she basically finds the optimum solution, and there's now very little exploration. So with just a few dozen trials, the bee gets better and better until she finds a route that is at least close to optimal. Now what we can also do is to track bees' entire lifetime in terms of their spatial whereabouts. And what we have here is basically the maiden flight, the first flight ever of a bee that leaves the hive. Okay, this bee has never seen the outside world before, and we've put a transponder on her back and track her flight. The blue dot is the location of her hive, and she now explores in different directions. This is all one single flight that we're seeing here. She intermittently returns to the vicinity of the hive, and the tracks fade in color so you can see what's current and what's earlier. The more distant ones in time are colored red. And you can see that she explores in various different directions her environment. This whole flight lasts over 2 hours. And I don't know if you can see my mouse. I guess not here. So what's one thing that I would ask you to remember is that on the bottom, on the top left, there's a forest edge that the bee visits briefly once during this first exploration flight. And that will become important a little bit later. Now, on day 2, the bee does one more exploration loop, shown here in green, and then she's found a valuable resource. And now exploration turns to exploitation. She's found that flower patch, and now for several dozen foraging bouts and several days, that bee does nothing but fly back and forth to that patch, bring back nectar and pollen to the colony, and go right back to this foraging patch. So now from being an explorer very early in her life, she's turned to a resource exploiter. And after day 6, there were a few days of rainy wind, returned to foraging. And for a few flights shown here in green, the bee still goes to that patch that she had visited and exploited for several days before the rainy break. And then at some point, halfway through one of these regular foraging bouts, the bee appears to change her mind and flies to a location that she had only visited a single time during her very first flight out of the hive, without retracing her steps back to the hive, and then continues exploiting a new patch for the rest of her days until bees have short lives. She was probably taken either by a predatory bird or a crab spider or something like that, and she just suddenly vanished from the radar. So there's a bee that has a very brief exploration phase in her life, and then spends the rest of her days exploiting first one then another patch. But what is especially remarkable about this bee's tracks was that while she visited only these 2 patches, is that along the tracks shown in this figure, at some point she appeared to change her mind and fly straight to a destination that she'd only seen once 10 days ago. Now, all this work that we did on spatial learning and flower color learning and so on, while we were impressed with it when I talked to colleagues working with primates and corvid birds and other glamorous big-brained animals, these colleagues were always a little dismissive. And they said, well, okay, they learn something, but what they're learning is just the stuff that they need to learn every day in nature. And they said, but we, with our crows and magpies and chimpanzees and so on, we deliberately face the animals with tasks that they have never encountered before. Now that would show real intelligence. And one of the tasks that my colleagues were using at the time was a so-called string pulling task, where you have to pull on a string to get a reward. And now here is a bee faced with a string pulling task. So this flower is an artificial flower with a nectar well in the center, and to get to that nectar well, you have to pull out the flower from under the glass screen to retrieve the reward. And the bee managed this just fine. Not only that, but you can actually pair a trained bee marked here with the red dot with an entirely naive individual, not marked with the red dot, and the uninformed naive individual will learn the technique from a trained demonstrator. So in this case, you can see 4 flowers, artificial flowers, each with a nectar well and each with a string attached. And so now the 2 bees are happily sucking the nectar side by side, but the naive bee has not done any work. They're simply scrounging on the efforts of the trained bee. Now they've emptied the well. You can see they're now getting a bit nervous because there's nothing left. But the trained bee runs straight over to the next flower and pulls the next flower out. The naive bee still thinks, there could be something here. But now she's running over to join the experienced bee. And now they're again sharing the nectar reward that's in the central well. The interesting thing is if you repeat this a few times, you pair these naive individuals with the trained bee, is that the naive individuals will themselves learn the technique. And in this case, again, now their flower is empty, they're running over to the next flower. You can see the naive bee is still a bit helpless as to what to do, but again, she now joins the experienced bee and so on. You do this a number of times, and the previously naive bees have learned this technique from the experienced bees. And what happens if you do that with a good number of bees is a kind of, is that the technique spreads through an entire bee population, like a meme on social media. What we have done here is we've arranged all the bees of a colony in a circle. Each dot is a bee. And so the first individual that was trained is shown with a yellow dot at the top. And once a line forms between any individual and another one, that's the first time of an interaction of the type that I had just shown you in the previous video. And after a while, these dots turn colored. The first time that they're turning colored means that the previously naive individual has now learned the technique. Okay? And so you can see now there's a good number of orange individuals. These are all the bees that have learned the technique from the initial single individual that was the inventor of the meme, so to speak, at the top. But now you can see at the bottom here there are a few purple dots. And these are second generation learners, not biological generations, but meme-wise learners, second generation, in that technique from the initial demonstrator, as we call it, but from the orange ones. So the purple ones have learned it from the orange ones, and the orange ones have learned it from the yellow ones. And the technique even continues to spread after the original individual, number 31 at the top, has passed away. Okay, so the technique spreads and spreads so long as there's value in learning it. And once you've trained a whole bunch of bees, this is slightly sped up, but then they sort of learn to work all of them together with pulling out a bunch of flowers to reach the rewards. This is quite entertaining, actually. But you can see that they prefer to work, they join each other on pulling on each of these strings until they've removed all of them. Now, the introduction for the next experiment I'd like to leave to Jane Goodall. I don't know, the older ones of you might know about her. She's not necessarily familiar to many of our younger students, but she is a famous primatologist who has pioneered work on the behavior of primates, chimps especially. And she's heard about our bumblebee experiments, and this is what she says. Now, the audio is a little bit quiet, so you'll have to sort of listen up a little bit.
The most recent example which blows people's minds is the bumblebee. And the bumblebee has been taught to roll a little ball. He rolls it backwards, rolls the little ball until he can push it down, or maybe she, I don't know, into a hole, like a goal. And as soon as this little ball gets into the goal, the bumblebee is given a little drop of nectar as a reward. So they learn to do this. But the mind-blowing thing is, other bumblebees who've merely watched a taught bumblebee can do the same without being taught, just by watching. And that is supposed to be a mark of very superior intelligence. So this is something we're learning all the time. We have been far too arrogant. The animal kingdom of which we are a part is filled with secrets. And gosh, I love to be young and learning about these things now, because all the doors are wide open and you never know what you're going to find.
Yeah, I was very moved by that Jane Goodall would hear about our bee work and say, gosh, I like to be young now and explore bees instead. But yeah, they are amazing. So let me explain a little bit more what this experiment entails. So after the string pulling work, we asked basically, can the bees move an unattached object to the location of reward in the same way as you'd describe tool use or operating a vending machine in the human realm. So the idea there is indeed that this ball belongs into the center and the bees have to learn to move it there, and there's one skilled bee doing exactly that. But and now she gets a little sugar reward from underneath this platform. But the way in which we trained the bees in learning this technique from observation involved a little trick. And the trick is, you can see that there are 3 balls here. And of course, the best way to solve the task of move ball into the center is to pick the closest ball, is to pick the one that's nearest the yellow circle at the center. Now, the trick that we played is that the experienced bee at the bottom left there actually knows that the closest ball cannot be moved because we've glued it down. Both the closest balls are glued to the surface, and that bee knows I can only move the furthest ball, the one that's furthest from the goal. And we paired again this trained individual with a naive one that had no experience with ball rolling, and this is what that untrained bee got to see. So there's an experienced bee that rolls the ball. Once it's in the center, they both get a reward. We do that 3 times. 3 times the naive individual gets to see the experienced one roll the furthest ball to the center. And that, of course, is, in theory, if you can move all the balls. But what we did then is we put the previously naive individual on the spot and ask her, well, I'm always saying she because these are invariably worker bees, which are all females. So we ask this bee, how would you solve this task? And the question there is, would she simply copy the demonstrator or ape the demonstrator, as people somewhat flippantly say, by moving the furthest ball, which is what she had observed and had been shown by an experienced bee, or would she solve the task in a better way? And invariably, this is what we find. The previously naive individual said she's very clumsy at rolling the ball. She's never rolled balls for rewards before, but she picks the closest ball. And we interpret that to mean that rather than simply copying the actions of the demonstrator bee, is that there is a kind of understanding of what the desired outcome is of these actions and solve the problem in a better way than the demonstrator had. So there's a kind of intentionality that requires the bee to, at least at some basic level, understand what the nature of the task is. Now, can bees imagine things? Can they picture objects in their minds? Now, we know obviously that they can learn flower colors and patterns and so on. Here is again our bee view, slightly distorted through a bee's eyes as she's scanning flowers. But it's actually surprisingly difficult to prove that there is a little mental image floating around somewhere in a bee's brain, because many of such recognition tasks could actually be solved with very simple feature detectors, with neurons that recognize edge orientations, colors in certain areas of the eye, and so on. There's not necessarily a need to memorize the thing in its entirety. And one way in which you can ask if an animal has a mental representation of an object is to ask whether that representation is accessible from multiple different sensory modalities. And so what we've done here is that we've trained bees to recognize certain shapes, either in the light, in a setting where they could see but not touch the object, or in complete darkness, where they could not see it but touch it. So that's the experimental setting shown here. There's a simple Petri dish on the left where the bee can see the balls through the lid but cannot touch it. But in this case, she needs to learn to associate the ball with the reward and to know that the cube is unrewarded. And in darkness, what you see is that their lid has now been removed. They can touch the object, and we can then make visible using infrared light what the bees are doing in darkness. And if we do that, so here, for example, we train our bees in light, can't touch the object, but can see it, then of course they learn to associate that. But we then transfer the bees into a setting where they can't now see anything. They just walk around in the dark. This, by the way, is part of a bee's natural behavior. So their nests are naturally in the darkness. So before they fly out, they operate by walking around and feeling their way around. They're highly sensitive antenna, for example, to sense their environment using just their tactile sense. In that case, they spontaneously recognize the ball as the rewarding object. And of course, we have control groups where they're learning the cubes as being rewarding. And vice versa, if we train the bees in darkness and then expose them to these types of objects when they can only see them but not touch them, they spontaneously again pick the correct shape, even though in the past they've only been associating them with rewards in the tactile modality, which seems to us to indicate that there is more than just simple feature detectors in just one sensory modality, but that the basic properties of the object are accessible from multiple sensory modalities. Now, perhaps to what most people would regard as the most speculative part, the question of whether insects have emotions and attach them in some way to spatial settings. And this was regarded plausible, actually, by a pioneer in animal and insect cognition, Charles Turner, over a century ago. He is an African-American scholar who at the time published his work under extremely difficult conditions. He was not able, because of his ethnicity, to find employment at a major research university, and did much of his work at a special school for African American children. Nonetheless, he published over 70 original research articles, among which were 3 in the journal Science, and often very visionary work on insect, but also other animals' behavior, that while he did manage to publish it at the time, was quickly forgotten and is now unfortunately not known to many people. But he said this about emotions in insects. So he took this photograph and he comments on it: the coiled antenna, the protruding mouthparts, and the general attitude indicate intense excitement. One who believes that insects have emotions will find much in the attitude of these 2 Ammophilas to support his view. So these are wasps, of course, in this case. Now, Turner was aware that in this case, this is a speculative exploration. In this particular context, he didn't back his suggestion up with experimental work, but he has lots of very elegant experiments on insect learning and memory otherwise. He's also a pioneer of exploring what is now called animal personality, so he looked at individual differences in the psychology of animals and so on. We can now study emotion-like states, at least with experimental paradigms that in some cases, as in this one, are borrowed from experimental settings that are used on vertebrate animals, for example, domestic animals, animals in farming, to ask whether they are happy animals or whether their settings are suboptimal. And essentially, the animals are asked in an analogy to a human kind of approach: is your glass half full or is your glass half empty? Which is a sort of proverbial way of saying, are you a pessimist or an optimist? In this case, the glass is 50% filled with water, so it's an ambiguous stimulus, and you can either say, oh, that's pretty close to a drink being gone and be all sad about it, or you can see, well, there's just still quite a lot in there, in which case you might be judged as being an optimist or in a positive emotional state. And that's the kind of experiment, that paradigm that we've adapted here for bees. So you can see at the top left, there is a little flight arena, and there are at the right side of that flight arena 5 little tunnels. One on the left is marked in blue, and one on the right is marked in green. Now, what the bees learn over time is that, let's say for one group, blue on the left is rewarding, and green on the right, there's never a reward. And so over time, bees learn this, of course, and we can then subsequently, after they've learned this, give them various ambiguous stimuli that are between blue and green, and spatially between left and right. And that's these in-between stimuli that you can see on the bottom left. And then ask them, how do you judge these ambiguous stimuli that are between what you know as being rewarding and unrewarding? How am I doing for time? Do we have about 10 minutes still?
Yes.
Good, all right. So after the bees have been trained, we can quantify how they view these stimuli by simply measuring the delay after which they're ready to accept these target stimuli. And the familiar blue on the left stimulus, the bee know there's always a reward. What you can see is they fly in a straight line to that target. Whereas the familiar non-rewarding stimulus, green on the right, they will faff about a great deal, perhaps for 2 minutes, and then they might say, oh, okay, I might as well give it a try just in case there's something, but there's a substantial delay before they're willing to accept that option. And we can then face it with, this is now our glass with 50% water, the ambiguous intermediate option, turquoise, and ask, well, how do you judge that stimulus? Does that look to you more likely as being rewarding or unrewarding? And what we find in that case is that the readiness with which they accept that ambiguous stimulus depends to some extent on what happened before the experiment. Some bees, we have given a little surprise reward, a tiny droplet of sugar water before they even entered the experimental setup, 5 microliters, and the control group got nothing. They just were held up for the equivalent amount of time before they entered the flight arena. This is the only data slide. I hope this is, I've made this an almost no data talk, so I hope it's easy enough for the non-experts. So what the data that are shown here show the response times for the rewarding stimulus left of the x-axis and the unrewarding stimulus at the right of the x-axis, stimuli that the bees had not had previous exposure to. And what you can see is that on the right, the bees sort of spend about 2 minutes before they're accepting green. If there's the familiar blue, stimulus is less than 10 seconds. So a huge difference between what they know is rewarding and unrewarding. The other delay times are somewhere in between. But crucially, the delay depends on whether the bee, before being tested, had this surprise reward or not. Surprise reward is the red curve here, compared to no reward before. That's the black curve. You can see that the entirety of the red curve for all the ambiguous stimuli is lower. So they're more ready, they're faster at accepting the ambiguous stimuli if prior to the experiment they've had a little positive surprise. And at least in line with what we know from vertebrate work, that is regarded as evidence for a kind of positive emotion-like state in the bees. We also further explored this. We found that that this state is dopamine dependent. So what mediates this positive emotional bias is the transmitter dopamine. And another lab by Jerry Wright and others has found the reverse. That is, if they subjected the bees to an adverse stimulus before being tested, the bees acted as if they were more pessimistic. Now, if we're asking questions about an insect mind, you might be skeptical, even given what I've all shown you, and say, well, but I mean, that brain is just the size of a sesame seed or a pinhead. How could it possibly achieve anything as complex as a mind? And indeed, a bee's brain is small. There's no question about that. The neuron numbers inside it are also relatively small if you compare them to the 85 billion that we have. But it's by no means simple. So this, by the way, is just a frontal portrait of a bee's brain. The areas shaded in various shades of blue are the visual processing centers, lamina, medulla, and lobula. This sort of old-fashioned telephone dial-like arrangement, AL, is the antennal lobe. That's the principal relay of processing olfactory information. And dorsally, you can see the mushroom bodies, which function as centers of multisensory integration as well as learning and memory. Now, while that brain might have only about 850,000 neurons, this is one of these neurons. Okay? Every single one of these neurons might have the structure as complex as a fully grown tree. This is one neuron that basically constitutes the reward pathway in a honeybee. This is work by Martin Hammer from the 1990s, with the cell body at the bottom and extensive ramifications almost throughout the entire brain, the antennal lobes especially, and the mushroom bodies. There might be as many as 1 billion synapses, connection points between neurons in a bee's brain. And if you consider that at least potentially every one of these is plastic and can respond to experience by adjusting the connective strength, the complexity becomes pretty impressive. And it's by no means a simple brain. Even with much simpler brains of Drosophila flies, for example, while we now are getting closer and closer to getting a complete connectome, the entire connectivity of all the neurons that are being and how they're connected in the fly's brain. We're still a long way from understanding the functionality in anything that would approach a comprehensive understanding. And this was recognized very early on by Ramón y Cajal, who was one of the founding fathers of neuroscience, and he studied amongst many other brains those of bees and some other insects in comparison with, in this case, fish and reptilians. And this drawing, by the way, on the bottom here, the large figure is a drawing by Ramón y Cajal of the bee visual system, where you can see the eye on the left and then 3 relays with multiple different types of identified neurons. It's amazing in its level of detail and differentiation, and shocking to see how little we've advanced actually in over 100 years. So I'll try my best to read this. It would be silly of me to read Ramón y Cajal in English. So I'll try in Spanish, but you're not allowed to laugh, all right? Poseen estos animales un sistema nervioso extraordinariamente complejo y diferenciado, y de una finura constructiva que raya en los límites de lo ultramicroscópico. Comparados los ganglios visuales y cerebroide de una abeja o de un caballito del diablo con los de un pez o de un anfibio, experimentas una sorpresa extraordinaria. La excelencia de la máquina psíquica no aumenta con la jerarquía zoológica. Antes bien, se reconoce que en los peces y anfibios los centros nerviosos han sufrido inesperada simplificación. Ciertamente, la sustancia gris ha crecido considerablemente en masa, pero cuando se compara su estructura con la del cerebro de los apidos y libélulos, se nos aparece como algo excesivamente grosero, vasto y rudimentario. Es como si pretendiéramos igualar el mérito de tosco reloj de pared con la de una saboneta, maravilla de finura, delicadeza y precisión. Como siempre, el genio de la vida, al construir sus obras portentosas, brilla en lo pequeño mucho más aún que en lo grande. I think that's a wonderful citation about bees' brains. So just to wrap this up, I think our work has shown that bees do have mental representations of space and of things in space, that they can learn spatial things by observation, and that they use, in this case, simple tools in a manner that indicates some form of intentionality. They also seem to have flexible access to autobiographical memory and a kind of idea of things that they want, and explore suitable solutions to get there in a manner that somehow, I think, indicates an awareness of the outcomes of their own actions. And also that they seem to attach, at least by the same criteria that we apply to much larger brained vertebrates, some forms of emotional states. That's that. So if you have enjoyed that, have a look at the book. It's my first book, so I'm super excited by it coming out in a few months. But yeah, that's all. Thank you very much for your attention.
Questions? I have the microphones. I don't know if you can answer 2 questions at a time.
Thank you, Doctor, for such an interesting talk. I have 2 questions, actually. So all the work that you showed us was on female workers. What about the queen and the male reproductive? I mean, they do other things, I guess, so they know other abilities.
Yeah, so I mean, the reason why we're testing mostly workers is because of their unique behavioral trait of being highly motivated to work for very extended periods. Now, in honeybees, the males are pretty much good for nothing except for mating. They do no work whatsoever for the colony. They don't participate in brood care, in feeding the brood, in building anything. They only mate. They still have to remember where their native hive is, because after mating flights, if they're unsuccessful, they return back to that hive. One of the very first experiments I ever did was to test whether they could, the male honeybees, drones, could remember color signals at their nest, at their hive entrance. And yes, they can. They learn to associate the color of the hive entrance between and the refuge of home. So you can train them, but they don't naturally visit flowers at all. Nor do honeybee queens, for that matter. Bumblebees are a slightly different story, the abejorros, because the queen founds the colony as a solitary individual. They're the only individuals that survive the winter. And so they have to juggle all the tasks that, initially at least, before they have workers, that in honeybees the entire colony does. So the bumblebee queen needs to found a colony, to build the first wax structures, to find food for the workers until the workers start helping her. And one of my former postdocs, Nigel Raine, who is now a professor at the University of Guelph, has explored their learning abilities as well, and they're fairly smart bumblebee queens. So are the males, because in bumblebees, unlike honeybees, the males also visit flowers to feed themselves. In honeybees, they can't even feed themselves. They need to be fed by the workers. So we haven't yet tested workers in these more advanced intelligence tests. Sorry, we have not tested males in these tests, simply because they're harder to work with, because once their tummy is full, they'll go, "Thank you very much, I'm done with your experiment, and I'll go to sleep now." But we don't have any reason to suggest that they can't, at least in theory, solve them as well, because at least in those tasks where we have tested them, that is learning colors, for example, they're as fast as workers.
And the other question, if I may, is does sound have anything to do with color learning or spatial recognition, or shapes it in any way?
Sound.
Sound.
Possibly, but we don't know. So unlike some other insects that communicate over very long distances with sound, like the chicharras here, honeybees don't hear very well. And much of the sounds that we hear in bee colonies are actually transmitted by substrate vibrations. And actual sound, that is airborne waves, are only perceived at very short distances, just a few millimeters to centimeters. And I guess because that was recognized early on, sound perception and its relationship with learning and memory is profoundly underexplored in bees. So we don't know is the honest answer. It could be, but it's not one of the sensory modalities where they shine and excel in comparison with other animals.
Thank you very much for. Can you hear me? Okay.
Yeah.
Thank you very much for the talk. I really enjoyed it, and I think it was a great restart of the seminar series in our department. So thank you for being here. Related, I have so many questions, but related to that question, I'm surprised. I work with Drosophila as well.
Okay.
And I was surprised that you didn't talk at all about olfaction.
Mm-hmm.
And I'm sure, I mean, all the learning cues you assume were visual, and in some experiments that you show, you talk a little bit about the tactile sense. So what about olfaction? So how does it play a role in these experiments? Can you tell us?
It doesn't play any role in these experiments because we were very careful at removing any kind of olfactory cues that the bees might leave behind, which they do if they're left to their own devices. They have smelly feet, and so wherever they land, there is a trace of their recently having been there, and those bees that have left these marks, as well as other bees, will use the marks. So, and if we don't clean them up, then the bees will just follow their own scent marks. So it's important to exclude olfaction when we're asking questions about visual targets, for example. I haven't worked with olfaction very much, again, for sheer convenience, because it's always messier than visual stimuli. The nice thing with visual stimuli is you can remove them and they're actually gone that instant. You have it on a screen, you turn it off, it's no longer there. Scents, of course, linger around for longer, and especially in these flight arenas that we're using, they're harder to remove experimentally. But many colleagues who are perhaps less lazy than I have explored olfaction in bees and its relationship to cognition and memory in detail. And so there are many exciting results. For example, Mandiyam Srinivasan has worked with training bees to multiple different destinations in their flight range, where they had to associate scents, olfactory signals, with locations. And not only can the bees remember both the locations and the scents, but they can also, when you then subsequently blow a scent that's only been associated with one location into the hive, and then ask the bees which location they can retrieve from memory given only the scent as a cue, they will fly to the correct destination outside the hive. So the scent there activates a memory of the spatial setting. And you can do this, of course, with multiple locations, so the bees have to associate one location with one scent and a different location with a different scent. So there seems to be a linking of spatial information with scent information that the bees can retrieve in situations where they've never experienced the scent before. So there's this sort of work. I had briefly mentioned the work by Geraldine Wright on negative emotional state. So we used visual stimuli, blue and green in this particular case. Jerry Wright and team actually used olfactory stimuli, that where one scent was associated with reward and the other with no reward. And then she gave them ambiguous stimuli that were mixtures of these two scents. And again, she asked how they would judge these ambiguous stimuli, and in this case, the stimulus to put them into a negative emotional state, she briefly vibrated the bees with a device that's usually used to mix liquids, and then set them onto her olfactory discrimination task in that case. So there's quite a bit of work on olfactory memories and cognitive aspects, including peak shift and so on, in bees by other labs, but not so much from us.
Thank you. And if I can ask another question, in the bee salesman problem, so you mentioned, when I saw the trajectories and how they get better and better when they try more and more, I see that they always start with the closest.
Mm-hmm.
Is that right? So is there kind of a limit where you can, so if you start putting these destinations like farther and farther away, I assume that there is one way that they cannot kind of reach the destination. So I assume that the way they can reach so far away in nature is they recognize these points in the path, they mark them somehow, with olfaction or visually, whatever, and then they start going farther and farther away.
Mm-hmm.
Is that right? Is there a limit? Like, have you explored that? And also, what happens if you remove one of the earliest destination? What happens?
Yeah. So that's a very good question. Scent marks, again, we have to make sure to wipe them off every time a bee has landed, because otherwise, yes, they will like very much to use them. The particular array that we used in this case with these 5 locations was actually designed to trick the bees. So our earliest arrays were the flowers were further apart, and actually so far apart that they could not see one station from the preceding station. So they had to memorize the flight vectors. In this case, the array of flowers that we set up was actually deliberately putting some pairs of flowers very close together so they could see them, and we wondered, would they then prioritize, as you say, short distance moves over optimal moves? And the answer was somewhere in between. So even though in this case they found the optimal route, they were to some extent tricked by the attraction of seeing a nearby flower station, which actually took them away from the optimal route, which would have been just around the edge in this case. So yes, there is having flowers in close proximity, I guess, tempts the bees to depart from the overall optimal solution, at least to some extent. We also did an experiment where we first had the bees learn a 5-flower array and then removed one station and moved it somewhere else. In that case, there was a very strong tendency of the bees to return to the location they had previously known as rewarding multiple times before they actually gave it up. So there was a strong attraction to that location that they remembered as rewarding, even though it wasn't there anymore. And that attraction to this previously rewarding site also to some extent delayed them discovering a new site. But they did, with some probability, but with a delay, because there was a strong tendency to stick with what was familiar.
Thank you, Dr. Lars, for this interesting talk. My question is more about curiosity. You tell us that the bees fly around the colony for know his place. And do you know what happened if the bees lost, another colony accept, or what happened with these bees? And thank you.
It's a good question. So in beekeepers' yards, where there are often multiple beehives next to each other, it's actually not necessarily a rare occurrence for bees to either by error or deliberately fly into neighboring colonies. The hosts of the inhabitators of such colonies don't necessarily accept that very readily. So every beehive typically has guards that antenate and using olfaction or contact chemoreception check whether the bee actually belongs into that hive. And so more likely than not, such intruders or straying bees might be attacked and repelled. But as everything in biology, that recognition system is messy, and so it's not rare actually that bees manage to settle in a new hive, especially in conditions where there's lots of floral food to be had. So competition isn't necessarily strong. Then the guard bees are more forgiving in a sense. Later in the typically in at least in temperate conditions in the autumn, when there's less floral food to be had, the guard bees become more vigilant, because such intruder bees not actually only just enter new hives because they've lost their way, but sometimes deliberately to steal honey from other colonies. And so the guard bees are quite keen to prevent that from happening, but it does happen. Honeybees can't survive on their own. So if they're completely lost and don't find either their own or another hive, they're bound to die. Bumblebees to some extent can survive even as solitary individuals. Mm-hmm.
Are there any more questions from the audience? Okay. Yes. Thank you for the talk. It was very interesting. And my question is, sorry for the noise.
No problem.
My question is regarding the structures in the brain of the bee, the mushroom bodies that are analogs of memory and learning structures in the mammal's brain. Do you think that there's a chance that some of the cells present, for example, in the hippocampus, such as place cells, are present also in these brains?
Possibly, we don't know that. It seems that so these brains, of course, evolved along very different evolutionary pathways, and there seems to be a lot of functional convergence, at least. I mean, many of the building blocks, molecular channel proteins and so on, exist both in the vertebrate and invertebrate lineages. The brains are assembled a little bit differently. It seems that most of the functions related to spatial memory and navigation are in the central complex, so a different area of the brain that also processes head direction, compass direction, and a kind of virtual reflection of the polarization pattern of the sky. And so it seems to be that area of the central complex that seems to be more involved with spatial navigation than the mushroom bodies are.
First, about the neurons in the brain. There is a lot of neurons in the eyes. Are there some information activity in the eyes, or are only receptors?
Yeah, you're right. So there is a tremendous diversity of neuron types in not just the eyes, but all the three optic ganglia, lamina, lobula, and medulla. That actually, in its diversity, is best explored in fruit flies again. But I don't have any reason that the diversity of neuron types is any less in bees. And it turns out from people's work at Janelia Farm and so on that the diversity of neuron types in the visual optic ganglia of insects seems to be higher than the human retina. So there are over 140 different neuron types in the optic ganglia alone. And many of these are recognized as feature detectors, such as overall brightness detectors, color opponent neurons, edge detectors, various types of motion detectors over either small or large visual fields, and so on. So yes, there's a huge diversity and quite a bit of processing in the visual periphery already that I guess in humans would take place to a large extent in the visual cortex, not the retina.
And the other, about the emotional stages of the bees, you show something more motivated and less motivated. Is this something related with hormones, or what is the basis of this difference?
So in our study, this, what we call the positive emotion-like state, seems to be mostly dopamine mediated. And that, I think, is backed up from, again, Drosophila work. So Scott Waddell's team in Oxford has looked at transmitters underpinning motivation. And again, it seems that the principal candidate there seems to be dopamine, as I guess in the mammalian reward system.
Okay, because, for example, in mosquito, when the mosquito is biting, he get a stage when there is a non-return stage. If he is biting, he continues biting even if you kill him. So this is part of a hormonal stage. So this could be, if this could be considered an emotional stage too.
It's certainly possible. I mean, we don't. I don't think this has been explored specifically in mosquitoes, but it's interesting. I think that while people are willing to acknowledge, for example, the unpleasant sensation of pain as a sentient state in mammalians, and the very fact that it can be modulated in mammalians and other vertebrates is taken as evidence for it being a mind state. The opposite seems to be the case in invertebrates. So often the observation that, for example, in praying mantids, where the female sometimes consumes the male in the process, people have cited that as evidence that there cannot be a sensation of pain. But what more likely this indicates to me is the very modulation of nociception or pain according to behavioral context. So when the stakes are high, when you have in theory something very high, a very high price to win, as for example the mating success in the praying mantid, and the mosquito that fills up one meal to secure nutrition for her offspring, then phenomena like pain, nociception, as well as escape responses might be adaptively downregulated. Now, humans, of course, are particularly dangerous hosts for mosquitoes because we can use our hands in a targeted way. The mosquito doesn't necessarily know that. So, for example, if a mosquito attacks a dog or a horse, then the worst that can happen is that someone swishes a tail at you, which is most likely not lethal. So you might as well stick with your task once you've already managed the landing safely. So I suspect that this irresponsive to an adverse stimulus in that setting is actually indicative of the very modulation of what would otherwise be a simple reflex-like response. But we don't know that in mosquitoes in detail. Okay.
Okay, thank you. A very interesting talk. According to your conclusions, and I agree with them, so intentionality, the exploring suitable solutions for specific problems, emotional attachment, and in particular, awareness of the possible outcomes of the actions, they point to, as you said at the last line, they are key ingredients of a mind. But they also are based in a very complex neuronal circuitry. And when the relationship between brain and mind is talked about, consciousness is always at the heart of the discussion. And that is my question. Is there any evidence, information about consciousness in bees? For instance, self-consciousness?
So that's what we're trying to explore at some level, but of course that question is as difficult as it is with any other non-human animal or indeed any non-human entity. So we humans as experimental subjects as well as experimenters have the huge advantage that we can communicate about our sensations, about our self-awareness, and so on. And that ends with humans. We can't do that in any other animal. We can't do it with artificial intelligence to the same extent. So there is not in any animal at this stage or in an artificial intelligence system an accepted decisive proof for something like consciousness. So everything we do is to some extent based on common sense and probability rather than decisive proof. And in that sense, so some of the approaches that we have used, I think, at least point in the direction of there being a so if you contrast a conscious entity with one that perhaps lives entirely in the present, with the historic view of animals and insects specifically, that they're just reflex machines whose diverse repertoire contains just innate routines that are triggered by one stimulus or another, then I think we already have evidence against that. We have evidence that they can access relatively flexible memories from the past, that at least at some basic level there's a projection into at least the immediate future and so on. Now that does not mean that they have the levels of consciousness that we have today as humans. So I would not suggest that a bee can reflect in the morning that, I'm somewhat in a bad mood today, so I'm not going to go out and visit flowers. So I don't think that level of being aware of one's own emotions, for example, is realistically part of a bee's spectrum of capacities. But there are interesting experiments by also other teams in this case. So there is one question of how consciousness is explored in other animals as metacognition, is the question of whether an animal has an awareness of the certainty of its knowledge. So let's say if you're facing with an exam task, for example, then there are some where you know, okay, I know this, it's easy, I don't have to hesitate to put down the answer. Whereas with others, with a bit of scratching your head, you can do it. But even after the exam, you might still think, well, was I right? Was I not right? So you have an awareness of your own certainty, of your own knowledge, a knowledge of your own knowledge. And this kind of task has been adapted to animals, including bees, by, in this case, Clint Parry and Andy Barron, where they asked if they gave bees, in addition to answering yes or no, is this the right stimulus or not, they gave them the option to opt out of a setting if the bees were not sure. And it seems that with increasing task difficulty, the bees were more likely to just say, no, sorry, I'm not sure, and fly away rather than making a wrong or a right choice. So there is at least some tentative evidence in the right direction, but it's not formal proof. But then again, for fairness, we don't have that formal proof for dogs or chimps either, or an artificial intelligent system. So for all of these non-human entities, we have to collect shreds of evidence, ideally from multiple different angles. But yes, a computer person might always counter, well, I could implement that in a machine as well. Our answer to that typically is yes, but that's because you've built this particular capacity deliberately into the machine. It doesn't naturally pop out of it.
Okay, now we're going to read out some of the questions from YouTube. I want you to know that there were up to 40 people connected to your webinar.
Okay.
And many nice comments and congratulations on the seminar.
Muchas gracias.
I'm going to read one question, then Ricardo will briefly take care of the rest. So Felix Torres Gonzalez is asking, how will we know if the brain of bees degenerate with age? Do they display something similar to human senile dementia? And then he says that solitary old bees get disoriented in their recognition of the hive. Is that correct?
Mm-hmm. So I don't know if bees' brains deteriorate with age. I think one thing that we have to keep in mind for all non-human animals that operate under the rigors of natural selection is that they typically die before they reach that sort of age where brains disintegrate. And if you imagine primordial humans, I mean, you presumably, at least I learned in school that just a few centuries ago, the average life expectancy of a human was somewhere in the 30s in most parts of the world. And so you're more likely to die from disease or predation or accidents under the rigors of natural selection than to reach the age where brain disintegration becomes a realistic threat. worker bees only live a few weeks, so there might be the wrong system to explore that. Interestingly, however, there are insects, and that includes in the bees, that live for many years, and in the bees, that's the queens. In termites, there are the queens can become up to several decades old. And so in that case, there might be an interesting case to study senescence of the brain, but I simply don't know to what extent that's been done. But it could be an interesting question because indeed they do age substantially, and of course they are also very protected from adverse influences, these insect royalty, termite queens and so on. I'm afraid I forgot the second question. What was the second question?
Solitary old bees try to forget how to recognize the hive. Is that correct?
That's a good question. So many solitary bees actually face extraordinary challenges. There are some wasps which are even more impressive in that they maintain multiple different nesting sites. And so they don't just have one home, but multiple. And I'm thinking especially of one species of wasp that over the course of the season will have up to 10 little nests, typically around 3 at a time, which they gradually provision. And then eventually, when they're fully provisioned, they abandon them and focus entirely on new nests. And so these bees, wasps in this case, not only have to remember the locations, but it seems that they remember how recently they have provisioned each nest and provisioning them and prioritize re-provisioning the ones where the last visit was the furthest in the back. But also part of this lifestyle involves eventually abandoning nesting sites which they have fully provisioned and then focusing entirely on the new ones. I'm not sure if it's known whether they entirely forget them or just know to disregard them because they're fully provisioned, but it could be that they just erase them from memory. That's also correct. Mm-hmm.
Well, good afternoon, Dr. Lars. I really enjoyed your talk. Do you have one question from Niels Schut in the audience? He wants to know what happens within a new hive compared to a hive after hibernation, and he sees some of the knowledge transfer, or does the hive starts again from zero?
Another very good question. So this depends again on the species that we're referring to. So temperate bumblebees, for example, start in every season de novo. So because the only individuals that survive the winter is the queen, and they then found a new colony entirely from scratch, and then it grows up to several hundred workers over the course of the season, and at the end of the season the colony collapses. However, there are some tropical species of bumblebees whose colonies persist for multiple years, and in that case there is certainly a possibility for the knowledge also to be transmitted over multiple seasons. In honeybees specifically, people have explored whether memories from the autumn are still accessible in the next spring. So what happens again in temperate habitats, unlike here, is that the hive shuts down for several months and there's no foraging for the winter months. And Martin Lindauer, who was one of the pioneers in exploring the bee dance language jointly with Calvin Frisch, asked whether these bees that spend several months locked up in the hive still remember spatial locations when they re-emerge from hibernation, basically in the subsequent spring. And the answer is yes, they still remember such spatial information. And to the extent that there's still forage there, they will also, using a special communication system that exists in honeybees, inform other bees of these locations.
He was wondering as well if pesticides affect these behaviors.
Yes, So pesticides, unfortunately, are typically the ones that we spray on plants, are actually designed to kill insects. They're deliberately made for that, and thus, unsurprisingly, they also affect bees adversely. Now, the bees don't eat the vegetation, unlike many herbivorous insects, so they're typically not killed directly, but they are exposed to various insecticides indirectly because they leak into the nectar of the plants, and they also occur in the pollen. So they will ingest them and carry them back at low dosages, which is now known to affect all kinds of things adversely. Even at a sublethal level, it does affect spatial memory, learning, it affects olfactory memory, and also navigation. So these pesticides are harmful at multiple levels, even if they don't instantly kill. And in beehives, of course, they are looked after by beekeepers, and they are therefore in a relatively privileged position because they're domesticated animals. So honeybees are not typically under threat. But there are also out there in nature, hundreds of thousands, in fact, of non-managed wild bees, and they, of course, are not rescued in case their colonies or solitary nests struggle, so they tend to be very much at risk from overuse of pesticides.
As Edmundo Lozoya as well was wondering if known if the learning of a bee may be transferred to another not trained bee by some biomolecules storing information like DNA or RNA?
It's not wholly impossible, but I'm not sure that there is a credible example of that. So it's interesting. So in the 1960s, I think there was a great excitement when it was found that when you transfer ground-up brains of planaria, certain memories seemed to be transferable to other individuals that were injected with sort of ground-up brains. And of course, at the time, this generated a vision of where at some point you would consume a little sort of pill and then learn a language instantly or something like that. And there was a guy, Professor Martin in Würzburg, who explored exactly that in honeybees and claimed that this also worked, that olfactory memories were transferable somehow chemically by him feeding ground-up brains to uninformed individuals. He received quite a bit of criticism and then spent the rest of his career stubbornly trying to prove it, decades long, and did not actually find any decisive proof. So it's not impossible, I would say, but I suspect that if it was a likely pathway, then we might know by now.
Okay, finally, Marcela Celis Santos has another question. It's, in your experiments, have you found situations in which you could say that bees were playing with what you offer, or out of an optimization or functionalist reading?
Yes. So one of my PhD students, Samadhi Galpayaj, has explored exactly that. So we found in these experiments where the bees had to roll balls to gain access to rewards, is that some individuals just seem to enjoy rolling balls around irrespective of whether we gave them any rewards. And it turns out that, as in other animals, this engagement with these movable objects peaks early in life for bees and then fades. So in that sense, it seems similar to the play behavior that's found in some mammalians. And Samadhi then also explored whether the activity of rolling balls could become a kind of unconditioned stimulus in a learning setting. So in that, she did experiments where she supplied where the bees experienced rolling balls in different color settings, color arenas, and she then asked whether, so the question there is the activity of moving balls experienced as rewarding in itself? And if so, can this become an unconditioned stimulus that in turn is predictable with a conditioned stimulus that surrounds the activity of playing? And that turned out to be the case. So they returned more likely to arenas the stimuli that were associated with arenas where they could draw balls. So there's at least some tentative evidence that this might be the case. But this is early days in this investigation.
Well, that will be all of the online questions.
Okay, and just to tell you that Fanny's was following your webinar, and he commented, "I cannot believe you didn't discuss bees playing England's and Mexico's favorite sport." Maybe next time. So I think at this point we will thank you for this great seminar, and it really sparked a lot of interest, great interest in our community.
Thank
and
you.
Let's thank Professor Lars for this talk.
Thank you very much. It's been a great pleasure. Very interesting discussion as well.
Well, after the audience squeeze all the wisdom of Dr. Lars, I thank all of you for attending to this seminar in person. And I thank also very much to the people who attended through the official YouTube channel of Simstar. That would be all. Thank you very much, Doctor.
Thank you. You've been a wonderful audience.